as you just heard, my name is Alex, and I'm working for Metrion Biosciences, and we are a contract research organization specializing in preclinical ion channel drug discovery. And briefly, we provide a variety of services ranging from high-throughput screenings using our automated patch clamp platforms and fluorescent plate readers, but we also do a lot of manual patch clamp and provide different assays. And we constantly develop and implement novel techniques to broaden our services. And today, I want to show you an example of how we use dynamic action potential clamp to characterize voltage-gated sodium channel mutations. So I'll give you a brief overview of the NAF1.2 mediated disease spectrum, give you a brief introduction into dynamic action potential clamp, and show you then a few examples. So whole exome sequencing nowadays continuously discovers novel disease variants, and therefore we need better tools to quickly investigate the functional consequences of such variants. And especially ion channels have been widely implicated in a number of human diseases, and this schematic really only highlights a few examples. And in general, such germline variations, they can occur spontaneously, rather than are called de novo mutations, or they can be inherited. They can be present germline or just be restricted to a special tissue, rather than are called somatic mutations. And as I just said, it's still important to functionally characterize such ion channel mutations in order to confirm that they indeed have a pathogenic role. And today I will focus on mutations occurring in SCN2A, which encodes the voltage-gated sodium channel NAF1.2. And I think today in the morning we had already an extensive introduction into sodium channels, so I'm not going to go in detail because they all roughly operate the same way and they have the same overall structure. So they're NAF1.2, it's widely expressed in the CNS. It's predominantly localized to the axon initial segment, and especially at early developmental stages, it's responsible for action potential initiation. And at the later time points, this role is then taken over by NAF1.6, and NAF1.2 then plays a predominant role in the propagation of action potentials. To date, there are a lot of mutations which have been identified in NAF1.2, and they pretty much occur in every portion of the channel. And they are in general associated with a disease spectrum characterized by epilepsy and autism spectrum disorder and intellectual disability. So generally speaking, truncation mutations and missense mutation resulting in a channel loss of function, they are associated with autism spectrum disorder and intellectual disability, whereas gain-of-function mutations, they are associated with different epilepsy mutations. But this is obviously, it's not that clear-cut, and there are exceptions. And also about 1/3 of these autism patients, they display seizures. In terms of the gain-of-function mutation, there seems to be a correlation with the severity of the variants and the severity of the disorders. So we have gain-of-function mutations causing a very drastic functional effect. They have been associated with infantile epileptic encephalopathy, which is a very severe disorder where epilepsy starts in the first year of age and then persists, and these individuals also show a severe neurodevelopmental delay. Whereas more moderate gain-of-function mutations, they have been associated with benign, often familial seizures, and the seizures also start very, very early on, so in the first year, but then they often stop by about 2 years, and the whole progression of the disease is much more moderate. And again, I can only stretch, it's still important to functionally characterize these variants, but not only to know, to confirm their pathogenic role, also when deciding therapeutic strategies. Because, for example, using an anti-epileptic drug specifically targeting sodium channels, that's indicated in the case of gain-of-function mutations, but probably counterindicated in terms of when you have a loss of function. And generally how we characterize such mutations is we recombinantly express them in a heterologous system, like I show on the Hexel, and then we perform a variety of voltage clamp protocols to investigate different gating parameters. But in order to get a good understanding of what it is these mutations actually do, we have to provide apply a variety of different protocols and look at a lot of gating parameters. And this is quite time-consuming, both in terms of experiments and analysis. And then very often, it's not very easy to interpret because these mutations, they don't fall always clearly into gain of function or loss of function, but they very often have features of both. And then this makes it quite hard to predict what this means in terms of physiology. And also, ion channels, they don't act alone. They are functionally coupled to other ion channels and transporters, and they will in turn modulate the properties of these channels. And while we conventionally always use our recombinant systems, they are easy to manipulate, easy to culture, they are useful tools. They just provide us a limited, they have their limitations in terms of studying their physiological consequences, as I mentioned. So now a better way potentially would be to use primary cells where these channels naturally occur, like in our case, cortical neurons. So we can study the channel in their natural environment, let's say, but then that's also not always straightforward because you have the endogenous counterpart expressed and you might not have access to knockout studies, and then that complicates the whole experimental design. So although like neurons, there would be a better physiological model, they are technically a bit more challenging to work with. And what we really want is we want to have a better readout from our recombinant cells. And that's why I want to show you in my next slides why we use dynamic clamp in order to kind of bridge the gap between these two systems to get a better idea of the physiological implications of such disease mutations. Dynamic clamp per se is not a very novel technique. It has its origin in cardiac electrophysiology and has then been afterwards widely applied to neuroscience. And in very simple terms, it's basically used to investigate the effect of a simulated, computer-generated conductance on a real action potential in real time. And for example, in this is not working anymore. In this schematic, we have a record from a cardiac myocyte, which is current clamped, and the membrane potential of the myocyte is continuously monitored and fed back to the computer. The computer then, the model calculates a current to be injected back into the myocyte, which depends on the actual membrane potential of the myocyte. This is now different. This is not the same as a simple current injection. Here we have two systems. They are in continuous communication with each other, and that's what makes it dynamic. So the current to be injected depends, thank you. So the current to be injected back really depends on the output of the membrane, basically the membrane potential of the myocyte at the given time. Then the current to be injected is adjusted accordingly and sent back. And this, as I said, is at a very high frequency. And that allows us to investigate this effect in real time. And this is now an example where they were basically investigating the effect of a so-called injury current, which would happen during a major cardiac event like a heart attack. And this then flows from the healthy tissue to the injured tissue and is shown to shorten the action potential. But again, this wasn't actually present. It was basically by incorporating this modeled conductance into the actual recording of the myocyte. Now what we do, we turn this story around. So we now investigate the effect of a real conductance by expressing our sodium channel variant in a CHO cell on the effect on a simulated action potential. Now, how does this work? So we have a model, a biophysically realistic model of the axon initial segment, and this model has been implemented by the group of Stephen Petrou, who is now the CSO of Praxis Medicines, which are our clients and collaborators, and they shared all this with us. And the model itself, in simple terms, is based on the Hodgkin and Huxley model, which basically says you can describe the action potential by the presence of potassium channels, sodium channels, and some leak currents, right? And that's basically what the model does. Now, what we do is we don't replace now these sodium channel conductances in the model by our actual recorded channels. So, our model, our computer model, it's in current clamp. We trigger action potential firing by in silico current injection. This will result that the model generates a membrane potential, puts it out, and sends it via the amplifier as a command potential on our CHO cell. At a certain point, the sodium channels in our CHO cell, they will respond, they will activate. Then this current is measured and fed back and integrated in the model. So basically what we are trying to do is, with this model, we mimic the presence of other ion channels as they would occur in a neuron. So that's what we are trying to do. And then, as I said, our currents, they get integrated, the model will change, the output, the membrane potential will change accordingly and be fed again back to the cell at a very fast pace. So, to summarize, this is an example basically. So this is here we expressed a wild-type sodium channel, did virtual in silico 10 picoampere current step injection, and this is an example firing what we would get. So, in a nutshell, what we're trying to do is we try to functionally couple a biological cell, our CHO cell, with a computer-generated model to create kind of a hybrid model. Someone called it the Frankenstein neuron, I think, yeah. And now I'm going to give you some example. So this is the first variant, RQ. Unfortunately, I cannot give you any more details about the position and the rest of the mutation. We did some classical voltage clamp experiments. So on the left, you see current voltage relationships, and you can already see that the mutation shown in magenta shifts the voltage dependence of activation, which then can also be shown by the corresponding conductance curve. And this is about a 5.5 millivolt shift to more hyperpolarized voltages. The voltage dependence of inactivation shown here was unchanged. So the fast inactivation kinetics, they were also slowed down by the mutation. And we also have a large non-inactivating component measured at the end of 40 millisecond, it should be, depolarization step at negative potential. So I think all of you would agree that this mutation shows clear features of gain of function. And indeed, when we did then our dynamic clamp experiment, we can see that the mutation increases the firing frequency over a wide range of current steps, and that's also shown here, and it also shifts the Rio base. Now I'm going to show you a second example. LQ, different mutation, similar shift in the voltage dependence of activation, also seen here, and this is now about a 5 millivolt shift, so before we had 5.5, now we have 5, and the inactivation is again unchanged. Now here, we don't really have a strong effect on the fast inactivation kinetics, but we have again an increased persistent current at negative voltages. So what do you think? Do you think these two mutations would behave very similar? Would you expect anything different in our dynamic action potential experiments? No? Same? Different? Same? Not quite, almost. It's still a gain of function, but it shows quite a different behavior. So here now, first, this is a different scale. So the amount of increased firing frequency is less, and it's only over a small range. So it behaves quite differently. And the point I'm really trying to make here is that with dynamic action potential clamp, if we ask, like in our case, does it affect neuronal firing? We don't need to interpret voltage clamp data, like, oh, is half a millivolt shift, a little bit of inactivation gating, what does this mean? We can apply this and we get a better readout, a better prediction what it might cause. And it might, in fact, allow us to dissect different severities. And also think now, I didn't have an example like this to show, but think you have a variant with a complex phenotype. Let's say you have both the inactivation and the activation shifted to more hyperpolarized voltages. It's even harder to predict what it's going to do. So this brings me to the end, and I hope I could convince you that dynamic action potential clamp can be a useful technique to characterize such sodium channel mutations, and it should give us a bit of a better physiological understanding, a quicker way to understand these mutations. And I mean, obviously, it's not all perfect. Everything has its downside. It's a model. A model to have their implication. And also, you can obviously only investigate the the effect of the electrophysical, physiological consequences. You cannot investigate any signaling effects, which might obviously also be changed. And there are only a limited number of models available. And I would like to thank our collaborators at Praxis Medicines and our academic collaborators at the Flora Institute, everyone at Metrion who contributed to this. You for your attention, and please don't hesitate to ask any questions.
So thank you, Alex. Do we have questions? There's one from here, please.
So, if I understood it correctly, using the Hodgkin-Huxley model, have you tried to validate that you don't get any changes in different nerve cells other than this model? Because you have, yeah.
If we did different, like, you mean, did we do different models or did we do actual experiments?
Different models for different nerve cells. Would you expect?
We only have this model. I don't know if I mentioned, but it's basically cortical layer 5 neuron. That's what it's based on, and the reason why we didn't test others is because they are not available. So that is a limitation. So if someone would write us the model, we would be more than happy to test.
Maybe check Allen Institute are getting out a lot. Allen Institute is getting out a lot of models for different nerve cells, I think.
It's good to know.
Are there any more questions? No? Okay.
Good.
Thank you very much, Alex.
Sure.